Hello, my name is Jose Cervantes, and today I'm going to be covering the credibility of magazine articles on Alzheimer's related research. The news story or magazine article that we're going to be covering today is a new method shows great potential for the treatment of Alzheimer's disease, and that was published by Science Daily website. The primary source comes from Uppsala University, and it's titled Enhanced Neprilysin Mediated Degradation of Hippocampal A beta 42 with somatostatin peptide that enters the brain. This is for Arizona State University Bio 282 lab, spring semester of 2021. Let's begin. So for the topic introduction, we know that Alzheimer's is a progressive neurological disease in which plaque forms between neurons and neuron receptors, which then inhibit neural communication and begin to gradually kill off brain mass and brain function. Now the plaque causing Alzheimer's disease, which will be abbreviated throughout the PowerPoint as AD, is known as amyloid beta peptide. Somatostatin SST is a neuropeptide that decreases or controls the amount of the amyloid beta peptide that forms around the neuronal membranes. SST cannot cross through the blood-brain barrier on its own because of its size, and therefore needs a protein carrier to carry it across the blood-brain barrier. And aside from that, it has a very short half-life. So that's why they're doing this type of research, so they can find a way to introduce it into the actual membrane where it's needed, instead of it just being in the blood and lasting around 3 minutes in there. So this is the basic overview of the neprilysin measurements. Now, in the graph right here, we have the SST on its own. That's the 100% control. Well, it's actually like a bit over 600. And then we have it when it's binded to this protein carrier. So SST would be the actual, what they're actually using to control the whole process. And SCFB FEBD3, that will be the actual SST binded to that protein carrier, and this would be the protein carrier on its own. And PBS is used as a negative control in this first part. So we can see here that SST on its own, we already know that it's very effective against controlling the production of the AB inside the brain. But we need this one for it to work. So the measures on this one, the gray one, would be about a 70% yield of what the black one actually, of the black bar actually is. And the protein carrier on its own, it doesn't really have an effect on the whole process. And PBS would just be, like I said, the negative control. This first part was for the primary neurons, and part B was for the SHS-Y5Y cells. So just a comparative measure between the both. Now for the magazine article, this treatment method, it specifically said this treatment method therefore reduces the formation of all types of aggregates, and it does, with the mention of all aggregates, it just fails to mention that the process only works when 3 consecutive IV injections of what the scientists have been testing out is performed, and not with a single injection trials, which were mentioned in the primary article. The primary source, by itself said no difference could be detected in the brain levels of AB40 and AB42, which are 2 AB derivatives. Neither in the soluble pool, figure 4C, which would be this one, nor in the membrane fraction, which would be this one. Between the SST, SC, the when it's already binded to the protein carrier, treated group, and the PBS injected controls. So the article basically just failed to provide that extra information. Sorry. Let's go to the next slide. The next slide would be the second part that the article missed on explaining the whole thing. So the magazine article said, in the study, the researchers saw the greatest effect in the hippocampus, the part of the brain that forms memories and the first part to be affected by Alzheimer's disease. Now, the part we're really focusing on right here is, in the study, the researchers saw that the greatest effect was in the hippocampus. When over here on this data, it's a little bit small to actually see, but we're focusing mainly on the dark one, which would be when it's already binded, because the rest are kind of, as we saw in the first part, they're just controls. And we can see that in some of the cases, like this one, the levels are a bit over outside of the hippocampus. And on some of them, it is greater, like on this one, you can see. So in this part, the issue was that the magazine article does not explicitly mention which effect of the hippocampus it is referring to, because it could be referring to the neuroleptin levels or the AB levels, which are caused by the neuroleptin levels. So it's kind of it's just not very effective at communicating the full information. Now for the next part. The overall representation of the magazine, I would say, is it was technically accurate with the representation of the primary source. The issue with the magazine article is that it kind of fails to represent the entirety of the study for full comprehension, as we saw that it only took the information from the primary source when it's talking about the 3 IV injections and not the first one, which in the first one, as we saw in slide 3 of the presentation, that was for the first trial. So in that one, it was not effective at all. There was no visible change in the levels of EB. So the first part actually failed when with the 3 injections is where Science Daily focused on without mentioning the rest. So technically the information is correct, but it's cutting out much of what it's really relating to. For credits, we have to thank Uppsala University for the publication of the primary source article. Here's the citation for it. The Science Daily website is what we have to thank for the magazine article publication. Also, here's the citation for it. And it's also good to know that this video was made in context of the Bio 282 conceptual approach for biology majors 2 at Arizona State University, spring semester of 2021. Thank you.